Alrighty, well, good afternoon, everybody, and welcome to the AgriFutures Chicken Meat Program webinar series. I'd firstly like to start with an acknowledgement to country. So AgriFutures acknowledges the First Nations people of Australia as the traditional custodians of the lands and waters on which we live, learn, and work. We pay our respects to elders past, present, and future elders of these nations and any First Nations people on this webinar today. In particular, I'd like to acknowledge the Ngunnawal people of Australia, the traditional custodians on the lands and waters where I am hosting this webinar from today. So a little bit of housekeeping about our webinar. We will be recording today's session and making that available through online channels. We have, so that we've got maximum sort of screen real estate of our presentations and minimize it and minimal disturbances to our presenters. We have turned everybody's videos and audio functionality off or on mute. And we kindly ask that you keep yourselves on mute so that we do minimize that background noise and distractions to our presenters. We will have time for questions and answers at the conclusion of presentations. And in the meantime, there is a dedicated Q&A chat function for you all to ask any questions over the course of the webinar. It is located along the, located, I think, along the bottom bar in Zoom of your screens. And we will be monitoring and responding to that chat function. So I'll start off with a bit of an introduction to myself before introducing our speakers. I'm Sarika Pandya. I'm the research manager, part of the AgriFutures Levied and Emerging Industries team, and I specifically look after the chicken meat research, development and extension program. One of the key research priorities within the program focuses on enhancing chicken biosecurity, health and welfare, and the program invests in research via industry levies within this area to support and strengthen industry biosecurity. This webinar is the second in a series in which we host expert presenters to provide you, our audience and industry with the latest information on topics that are relevant and also relate to our industry research priorities. Today's webinar is all about avian influenza, and it's designed to share and update the poultry industry on the evolution of the disease, including any developments that have led to the current panzootic, as well as a current research project to better understand why H5 low pathogenicity avian influenza circulating in Australia has the ability to convert to highly pathogenic forms. I hope you enjoyed today's content and find it informative. And with that, let's dive straight into today's presentations. Our first speaker, Dr. Michelle Wille, is a virus ecologist based in the Centre for Pathogen Genomics at the University of Melbourne, and with an honorary position at the World Health Organization Collaborating Centre for Reference and Research on Influenza. Her research focuses on the ecology and evolution of viruses in wild birds. She has worked on avian influenza viruses over the past 15 years and based in Canada, Sweden, and Australia. Beyond avian influenza, she has also addressed ecology and evolution of avian paramyxovirus and avian coronaviruses, and more recently has leveraged virome-scale sequences to reveal entire virus communities in wild birds. And with that, I'd like to hand it over to Michelle.
Thanks very much for the invitation, and going to be talking to you today a little bit about highly pathogenic avian influenza H5N1, so what's going on elsewhere in the rest of the world, and talking a little bit about some of the work that we've been doing to try to understand how these viruses might arrive here, and really trying to think about this in the context of poultry. But before I launch into that, I kind of just want to start a little bit with basics and talk a little bit about avian influenza viruses, what they are and how they're characterized, because these different kinds of viruses carry with them different risks. So on the left there on this slide is a little sort of 3D picture of avian influenza or influenza A. And these viruses are generally characterized in two ways. So the first one is called the subtype. So during the COVID pandemic, we learned a lot about the importance of the spike gene on the virus that causes COVID. But in influenza, this is kind of divided into two, and it's called the HA and the NA. Now, these viruses are very ancient. They've probably been in birds since birds have been birds. And so there's a lot of diversity. And so the HA, there are actually 16 different subtypes found in birds. And the NA, there are 9 different subtypes, and together we use these in combination. So we're going to be talking about H5N1 today. But of course, here in Australia, we've been learning a lot about H7N3, H7N9. Okay, so those are just the combinations. Within some HA subtypes, we further divide them into what we call clades or lineages. So this is sort of similar to the idea of variants in COVID, but we're not as good as COVID in coming up with nice names like alpha, beta, Omicron. Instead, we're using this horrific numbering scheme. And so the virus that we're quite concerned about, that I'm going to be talking about today, is this clade 2.3.4.4b. Okay? Not all HAs have sort of these associated lineages and clades, but H5N1 certainly does. The other way we divide viruses is by pathotypes. Now, the vast majority of avian influenza viruses out there are deemed low pathogenic or low pathogenicity. These viruses are actually quite common in wild birds in Australia, everywhere else in the world, predominantly water birds, so birds associated with water. In wild birds, these viruses don't cause clinical disease. We can go and sample and have 20% of the birds they're positive, and the ducks are just carrying on with their ducky things. The great concern, however, is when H5 or H7, low pathogenic avian influenza, enters poultry production systems. It's within poultry that they evolve to become more virulent or more pathogenic, and these are referred to as highly pathogenic or high pathogenicity avian influenza. Now, generally, these high pathogenic viruses are controlled very rapidly through a system of stamping out, which is what we've seen here in Victoria and New South Wales, with the exception of this virus called Goose Guangdong lineage H5N1. This virus entered poultry in about 1996 and has never been controlled, and that is the progenitor of this clade 2344B H5N1 that we're going to be talking about today. Previously, when I made this slide a few years ago, there used to be a little added bullet point here that said these viruses were uncommon in wild birds and largely restricted to poultry, but the situation certainly has changed, and hopefully I'm going to talk you through that. So what is the situation that we're dealing with? So in Australia, we actually have quite a good track record at dealing with high pathogenicity H7 viruses. As I said before, these viruses go from low path H7 into poultry. There they've evolved into high path, and they may sort of circulate within poultry. And generally, the approach of stamping out in poultry is highly effective because the viruses tend not to go from poultry back into wild birds, and we don't see the circulation of high path H7s in wild birds. But this 2344B H5N1 story is completely different and is actually quite a nightmare. I've made you just a little schematic here to try to capture the situation, but it is in fact a bit more complex than even this. But essentially, we're seeing a virus going from wild birds into poultry, circulating in wild birds, circulating in poultry, going back and forth. From wild birds, it's going into wild mammals, particularly marine mammals. In marine mammals, it's circulating within marine mammals and jumping back into wild birds. Viruses from poultry have entered dairy production in the US. They're certainly circulating in dairy cows. And they're in Colorado, particularly, we've seen these viruses jump from dairy cows back into poultry. And of course, both poultry and cattle viruses are causing human infections. And so the approach of just stamping out in poultry is not going to be effective and has not been effective against this virus anywhere in the world because the virus is circulating in so many different reservoirs. And so we really need to think of a new approach to deal with this virus when it arrives in Australia. So the global situation is quite concerning. So this is a map showing you the notification data of this virus since about mid-2021, which is when the panzootic essentially began. I've sort of colored here dots in poultry and wild birds, and you can see that wild bird and poultry outbreaks have been occurring essentially everywhere in the globe. The arrows there are meant to show you how the virus has moved and dispersed. And so essentially in 2021, Europe as the epicenter, we saw the virus move over the North Atlantic and into North America, and then it rapidly spread across North America. Since the index introduction in about 2021, there have been at least 2 additional introductions into North America, another introduction over the North Atlantic and one over the North Pacific. Following widespread sort of movement across North America, about 1 year after the index introduction, it entered into South America. We know there have been at least 3 introduction events into South America, but only one was successful. This virus then traveled the 6,000-kilometer spine of South America in approximately 6 months. That is a very, very fast-moving virus. Sort of reaching Patagonia in early 2023, and then sort of moving back northward along the Atlantic coast. And we're now seeing outbreaks sort of all across Brazil. About 1 year after entering South America, the virus was first detected in the subantarctic, and a few months later in Antarctica proper. The only part of the planet that remains free from this virus is Oceania, so Australia and New Zealand. This virus is having a massive impact. I realize this talk is about chickens, but really chickens and wild birds and wildlife are really linked in this outbreak. So when we look at the wild birds it's affecting, it's not just a few species, which is what we've been used to before. This virus has actually affected more than 350 species of wild birds. We did that analysis about a year ago, and it's certainly out of date now. So probably more than 400 different species of wild birds have been impacted. So the little plot there is from sort of a paper looking at the change in the number of species that have been affected since about 2005, which was the first time these Guangdong lineages jumped into wild birds. And critically, you can see since 2020, the bars are higher, the colors are fatter, and the red line is higher, just showing the massive increase in the number of birds it's affecting. In terms of wild birds, it's also having really devastating consequences. So these birds are not only the movers of the virus, they're absolutely the victims. Here is an example from South America. 40% of all the Peruvian pelicans in Peru died in about 2 months. That's almost 1 out of every 2 individuals from this species that have been wiped out from that country. In addition to wild birds, we're also seeing this virus in wild mammals. So the left there is just sort of an image showing you that everywhere we've been looking for this virus in mammals, it's been detected. It's been detected in at least 50 species. The vast majority of mammals that have been affected are predators or scavengers, which is not surprising. Imagine you're a little fox, times are lean, times are tough, and suddenly you appear on location and there's all these dead birds in the landscape. Wow, an all-day buffet. And you munch on all of those birds, and that, of course, leads to infection in these mammals. The clear exception here in wild mammals has been marine mammals, where in South America there have been almost 50,000 dead marine mammals. And my colleagues from South America are fairly convinced at this point that we are seeing mammal-to-mammal transmission in these animals, and mammals are giving the viruses back to birds. So there's been a real big shift in the epidemiology of this virus in South America. Going back to talking about poultry, this virus has had an enormous impact on the poultry sector globally. Just sort of 2 tidbits here. So in 2022, across 67 countries on 5 continents, over 100 million chickens were culled. That is only 2022. There is no evidence that the number for 2023 is any less, and that we would expect less in 2024. Indeed, in just one country, the USA, more than 100 million chickens have been killed since this virus entered the US, which was essentially around January 2022. So that is in 2.5 years, they've had to kill 100 million chickens. This is an entirely different scale to anything that we have ever seen before in Australia. The H7 outbreaks that we've been having right now, you know, people are talking about, oh, for 1 million chickens culled, that's a big deal. That is nothing compared to what this virus has done to countries and the poultry industry everywhere else in the world. Now, the US is a funny place because in addition to poultry, they've actually had it jump from poultry into dairy cattle. It's been detected in dairy cattle in 13 states, over 195 farms. These cattle are mostly getting mastitis, so they're getting really significant infections of avian influenza in their udders, and that is leading to huge amounts of virus in the milk. And the impacts of the virus in the milk are multifold. So essentially, we're having dairy workers being exposed to this milk, being infected. Part of the milk is apparently quite commonly given to barn cats in the barns. And so we're seeing a lot of dead cats in the barns. And in Colorado, they've actually now shown that the virus has gone from cattle into poultry. So we've got a situation where we had a virus in poultry go into cows evolution in cattle to sort of improve mammal adaptation, and then it's jumped back into poultry. And while the situation is still a bit unclear, there have now been a number of poultry workers in Colorado also infected with this virus. So we are looking at a really complex issue here of not just wild birds, not just poultry, but sort of at the farm scale here, interacting with wild birds. So very complicated. Now, I've alluded to human cases, but despite the enormous number of animal cases, there have actually been very few human cases due to this virus. So on this map here, the little dots in the background, that's from the map I showed you before. And then the little blue humans are the ones that are due to this 2344B H5N1. Now, in addition to the little blue humans, you can actually see that there's a number of other colored humans. And that's because in addition to clade 2344B H5N1, there are actually a number of other clades of this virus that are circulating, particularly in Asia. And these are similarly causing human cases. Now, largely, almost all the human cases have been in occupationally exposed people, so poultry workers, people going in to depopulate poultry farms. In Asia, people who work in live bird markets or people who buy chickens and visit live bird markets, people who have sick backyard chickens, and of course in the US, the dairy workers. There are one or two exceptions, but this is the general pattern. Now that's what's happening in the world. What are we doing about it? And what have we learned? And what are we thinking about here in Australia? So I just kind of want to just start off and recognize that the work I'm going to share with you has been done in collaboration with Marcel Claassen, who's at Deakin University. Since 2020, we have now collected over 19,000 samples from wild birds to be tested for avian influenza to better understand how avian influenza dynamics were circulating in wild birds here on the country. If you're interested in the work we've done so far, we've had a scientific paper come out not that long ago. And I just put a little map up there to show you sort of some of the sampling locations or some of the locations that we're sampling from, although that map is reflective of only the first 12,000 samples. So we've had a busy few years. Now, we've really spent the last 2 years really thinking about all the work that we've been doing in low pathogenicity avian influenza viruses. So those are the viruses that circulate naturally in Australian wild birds. And to help us to try to contextualize what might happen if high path arrives here. So we've been thinking about which species are susceptible to these viruses, how do viruses arrive in Australia, what drives the prevalence and dynamics in Australia, and how the viruses move and disperse within the country. Now, this talk is short, and so I can't talk about all these things, but I just want to touch on 2 points very briefly. The first one is how viruses arrive, and the second one is a little bit about what drives prevalence. Now, there's lots and lots of wild birds here, and not all wild birds are equal in sort of the role they play in avian influenza dynamics. And so what I've shown you here is a phylogenetic tree. This is a tree of relationships of sequences that have come from birds. And I've particularly put it on a time scale here, so that gives you sort of an idea of when key events happened. Now, the different branches are colored by different locations. So viruses from Asia and Europe are there in blue, and the ones from Australia are in pink. Now, we've been out sampling different kinds of birds, in this case, ducks and shorebirds, and we've been looking at the viruses that we're pulling out of these birds, and we're seeing a really clear differentiation in the role that these birds play. So if we look at the viruses that we get in ducks, so that's next to the little duck figure up there, we find that those viruses are most closely related to other sequences of ducks in Australia. So if you actually look at this tree and you look at the pink branches and you keep going back all the way to the left, right to where the box is, and you go all the way down, that's about the date that these viruses entered Australia. So looking here, the late 1970s. So essentially viruses from ducks are just circulating here, and the ducks are just reinfecting the ducks, and they've been doing that for decades. This is in contrast to the viruses that we're pulling out of shorebirds. Now, shorebirds are pretty amazing. This picture here, this is of a bird called a redneck stink. It weighs about the same amount as a tim tam, and this little tim tam bird flies from Australia to Asia and back every year. And the viruses that we're pulling out of this bird, sort of shown here in the pink on the bottom, are most closely related to virus sequences from Asia. So in this case, if you look up there at the end of the blue tips, you see South Korea, you see Vietnam, you may see Mongolia or Japan. So that suggests that these viruses are being infected or these birds are being infected elsewhere on their migrations and are bringing viruses from Asia with them into Australia. And this information is actually really quite critical when we try to understand which birds may bring high path into Australia. And so we discussed this quite at length in a risk assessment that we did for DAF a few years ago. The entire risk assessment is unfortunately not available, but there is an abridged version of the risk assessment that is available. And I put the big QR code there if you're interested in having a look. Now, I'm not going to talk about all of it, but I just want to go back into thinking about which birds may be bringing the virus here. So based on the study I kind of highlighted before, in addition to a lot of other work, we've identified that these shorebirds that use the East Asian Australasian flyway, as well as seabirds, may be responsible for bringing the viruses here. So how the figure works is the little left panel there. That's just the map of notifications I showed you before, just kind of zoomed in. And I just wanted to note there that Indonesia is a country that does not actually report any data. So the fact that there are no dots in Indonesia doesn't mean there's no high path there, it just means we don't know. The second figure there is just to show you all the places that Australian redneck stints and red-eared turnstones that we have captured and put flags on have been seen elsewhere in the world. So that's what those colors there. And if you squint a bit and you take the left map and the right map and you try to overlay them, you can see that a lot of the dots are in the same places. So these birds that are using this flyway are stopping in places where we know high path outbreaks are occurring, and they're arriving into Australia, millions of them each spring. Seabirds is a group of birds we actually don't have a lot of data on from a sort of a virus prevalence standpoint, but we do know that these birds are spending the non-breeding period or our winter in the Northern Hemisphere. And we do know that about a year ago, there was a positive detection in a short-tailed shearwater in Alaska, coinciding with a time period of when they would be leaving Alaska to come towards Australia. So this is another group of birds that we're really thinking about. And so in response to this, Marcel and I have designed an early warning surveillance system. We have run this program in spring 2022 and spring 2023. And at this point, we're still trying to raise financing to run something similar to this in 2024. But essentially in the spring, mostly around October, we're going to as many places in Australia as we can where we know that significant numbers of wild birds are arriving. And we're trying to catch and sample as many of these arriving shorebirds and seabirds as we possibly can. So Broome, for example, in the top left there is known as the shorebird capital of Australia. And it is the first sort of stopping point for a lot of shorebirds. Many of them may stay in Broome for the whole summer, but many of them will stop in Broome first and then come to the rest of the country. And so we're really hoping to continue this work again this year because early morning surveillance is really critical for us to be prepared. The last thing just to that I just wanted to touch on before I ended is just thinking a little bit about the factors that drive virus prevalence here in Australia. And so we've looked at lots of things. So we know age is important and species is important. Time of year is important, those kinds of things. But one of the things we really noticed in our data sets is that year is a really strong predictor of whether virus prevalence will be high or low in wild birds. And you might say to me, Michelle, that makes no sense. Duck fashion is exactly the same one year compared to another year. But the reality is we all live in Australia. We know that there are some years that are extremely wet and some years that are extremely dry. And if you were a duck yourself and you think like a duck and you are in an environment like the top picture there on the top right, you may say, why on earth I'm a duck? Why would I breed this year? I've got nowhere to build a nest, nowhere to lay my eggs. And I've got nowhere to take my ducklings to feed. This is in contrast to the picture there on the bottom right. You're a duck, you may say, oh man, conditions are fantastic. I'm going to go and build a nest and lay eggs and oh, look, I've raised all my chicks and conditions are still good. Let's try again. And so what this means is that in wet years, there are lots and lots and lots of ducklings. If you look at the population, most of those birds are ducklings. So the proportion of the ducks that are susceptible to avian influenza, that means they've got no antibodies, is enormous in wet years relative to dry years. And so when we do a correlation, looking at when influenza prevalence is highest in ducks, it is, of course, in these wet years where they can breed a lot. Now, this relationship is not only present in wild birds. When you look at poultry outbreaks, you also see that there is a relationship between rainfall and poultry outbreaks. Indeed, approximately 1.5 to 2 years after massive rainfall events, there is an outbreak of avian influenza in poultry, and the most recent outbreaks are no exception. So when we went back and looked at rainfall data, which is that little map, that little figure down there in the bottom right, you can see there is a really, really big spike at the end of 2022. We're about 1.5 years after that. So all that rainfall has led to lots and lots of ducks breeding. With a big susceptible proportion of the population, we're starting to see more and more low path infections in wild birds. And these wild birds sort of are then landing on poultry production areas and facilitating spillover, okay? So the plight of wild birds and the plight of poultry is very, very much linked here. And we know that, and it's not just here. And this is important for sort of these H7 outbreaks that we're good at dealing with, but it's also really critical for high path H5N1 because it is likely to be wild birds that are going to bring the viruses here and move them around the country and introduce them into poultry production. I'm just going to end with a plug for my resource page. This is my labor of love. I update it about every week, and everything that you might want to know about avian influenza is there. All the latest guidelines, all the latest risk assessments, the global situation updates, everything that's going on in cows and humans, and all the latest science. I made it to be a resource, so I really hope that you find it useful and use it. And I'm going to stop there. Thank you very much.
Thank you very much for that, Michelle, and thanks for that presentation, which provided an overview around that over avian influenza and providing those deeper insights into the disease, both in wild birds, poultry, and in other species too. For everybody on the webinar, in the chat function, we have put in some my support team has put in some links to some of the QR codes and resources referenced during that talk, so that you can easily click on those and view them. So now we'll move over to our second speaker for today, who is Dr. Jasmina Luczo, who is a virologist and team leader of avian infectious diseases research at the CSIRO, Australian Centre for Disease Preparedness. The mission of this avian infectious diseases research team is to increase Australia's preparedness against endemic and exotic avian infectious diseases through research that delivers a deeper understanding of disease pathogenesis, host-pathogen interactions, and virus evolution to inform the development of improved diagnostics and vaccines. Jasmina's research interests include avian disease pathogenesis, virulence determinants, antigenic drift, vaccine development, avian ex vivo models, and the functional evolution of avian viruses. Thank you very much. And with that, Jasmina, I'll give you the floor. Oh, Jasmina, you're just on mute.
Try again.
Yep, we've got you.
Perfect. Okay, is my screen sharing? Yes. Wonderful. Okay. Thank you very much to AgriFutures for the opportunity to present today. So I'd firstly like to begin by acknowledging the Wadawurrung people as the traditional owners of the land that I'm presenting from today, and pay my respect to their elders past and present. So today I'm going to talk to you about a project that's just commencing, that's supported by AgriFutures, and that's understanding the biosecurity risk posed by Australian-lineage H5, low pathogenicity avian influenza viruses, and the potential genesis to highly pathogenic forms. So avian influenza viruses are ubiquitous in wild birds, such as ducks, shorebirds, and gulls. And so in these species, the disease infection is subclinical, and upon spillover to chickens, it can produce mild disease. So we refer to these viruses as low pathogenicity avian influenza virus. So as Michelle nicely described before, we have 16 subtypes of avian influenza virus, So if you look at the top right, there's a schematic of the virion there. And so in the box is the hemagglutinin. So that's actually what we're using to subtype the virus. So we have 16 subtypes. And below that schematic is an actual micrograph of H7 avian influenza virus. And you can see that the arrows are pointing to that hemagglutinin, which projects from the surface of the virion. So in general, low pathogenicity avian influenza viruses cause subclinical or mild disease. However, there are two subtypes which are problematic. And these subtypes are the H5 and H7. And this is a subtype where high pathogenicity emerges. And this is a particular problem for our poultry industry.
Excuse me, Jasmine, sorry to interrupt. I don't think your slides are moving across for the viewers. Oh, now it's just shifted over to the correct slide. So I think we're with you now.
We're good?
Yes.
Okay, please do let me know if it's not working.
I don't think you're in full, I don't think your slides are in full screen either, Jasmina.
Yeah, it's...
Just jumping in here as the assistant Zoom manager here. So what you'll need to do, Jasmina, is just close your presentation, and then you'll have to open up the different screen where you have the presenter view.
All right. Oh, hang on. View. Sorry, everybody. Presenter view.
So it should be
okay. It's So I currently have it in presenter view now. Is that working at your end? Is it sharing?
Not as yet.
Okay, so try again. All right, so I have it in presenter view at my end. Is it at presenter view at your end?
No, it's not. So what you might need to do is just for now, we might need to just have you click on the slides when you're jumping through, if that's okay, on the original PowerPoint presentation page.
Sure. Okay.
Sorry about that.
Yeah, that's okay. All right, so you can see them now. I can just minimize that one.
Fine. That's fine. Thank you.
Sorry, folks. Sorry about that. Okay, all right. So let me get back into my train of thought. Okay, so high pathogenicity avian influenza viruses of the H5 and the H7 subtype do pose a problem to our pond spillover to poultry. Sorry, I'm having some technical... there we go. Is that working for you now? You can see the next slide?
Yes, we can. Thank you.
Wonderful. Okay, so highly pathogenic avian influenza viruses emerge from low pathogenic precursors. So on the left here, we have a schematic of low pathogenicity avian influenza virus infection in a chicken. So infection in a chicken is generally restricted to certain locales. So this is the gastrointestinal tract. It can be the respiratory system, the reproductive system, and the kidneys. But the replication of the virus is generally confined. Whereas highly pathogenic avian influenza virus can replicate in these systems, but also additional systems. So it's widespread systemic replication. So that can include replication in the heart, the brain, the spleen. So it does replicate everywhere, and it leads to severe disease and ultimately poor outcomes and death. So what causes this high virulence, this high pathogenicity? So the virulence determinant for avian influenza virus, major virulence determinant, is the hemagglutinin cleavage site. So again, we have the virion on the left with the hemagglutinin in the box, and then in the middle is a model of this hemagglutinin. So it's showing the structure of the protein. And I'm going to draw your eye to the bottom right of that middle structure, and there's a loop that's projecting. So that's where the cleavage site is located. And why is this so important? So monobasic cleavage site. So these are what are present in our low pathogenicity viruses. And these are cleaved by host enzymes which have restricted expression. So they are restricted to the GI tract or to the reproductive tract. Whereas we have these high H5 high pathogenicity viruses, which in this cleavage site motif contain multiple basic amino acids. And what that facilitates is cleavage by a different enzyme, and this different enzyme is actually expressed systemically. And what that facilitates is that widespread systemic replication that we observe upon infection with high-path avian influenza viruses. H5 highly pathogenic avian influenza virus, as Michelle has just described earlier, is a global threat. So since there's a table here that's describing the H5 outbreak since 1959, the first column is the year, the second column is the subtype. So we have the H type, and in combination with an N type, we have the lineage. So there are several lineages of H5. Michelle was describing the goose Guangdong lineage, but we also have a Eurasian lineage, we have American lineage, we also have these unique viruses in Australia that we call the Australian lineage. So high pathogenicity has emerged in every lineage except the Australian lineage. And interestingly, if you have a look in this table, I've bolded certain outbreaks, and H5N2 high path viruses are responsible for 56% of these outbreaks. And further to this, 8 of 9, since 1994, 8 of the 9 non-goose Guangdong lineage H5 outbreaks are caused by these H5N2 high paths. So that's an interesting little observation there. So we do see these highly pathogenic H5s emerging naturally. Then also in the laboratory studying it, we can also see the emergence of high pathogenicity in the laboratory. And so these are some core research that's occurred in the literature that's described the emergence of high pathogenicity and hitting on the H5N2s as well. So to date, what's happening in Australia? So to date, Australia has had 11 H7 high path outbreaks, and they're all described in the table to the right. However, there's not been one H5 high path outbreak, and the reason for this is unknown. And there's been lots of questions as to why, and one of the questions I do get is there no H5 viruses circulating in our wild birds? Well, through studies that Michelle's undertaken, we know that H5 and H7 low paths are circulating in our wild birds. So it's not because of a lack of low path H5s in our wild birds, resulting in a lack of H5 outbreaks. So there remains this overarching question and a key knowledge gap in the field. Why has Australia only had H7 high path outbreaks, but no H5 outbreaks? What is the biosecurity risk posed by Australian H5 low path avian influenza viruses? And ultimately, can our Australian H5s convert to highly pathogenic forms? So there's several unknowns about our H5 low paths that are circulating in wild birds. Can Australian H5 low path actually infect and replicate productively in chickens? In what tissues do they replicate in, and how efficiently does the virus replicate? Is it that the virus is able to replicate, but just replicates so poorly that the infection peters out? Is that the reason that we don't see that opportunity for H5 high path to emerge in Australia? Also, upon replication in birds, can we detect any changes in the genome with high pathogenicity? So, to address this, we'll be undertaking a study where we will do an infection study with our Australian H5s. So to determine which viruses to select, I generated a phylogenetic tree. So this is a simplified phylogenetic tree of H5 low path viruses in Australia that have been published. And so on the left, we can see that it is the relationship of the viruses over time. So on the left, we have our viruses from the early 70s, then we see the 80s, and then it progresses over time to our contemporary viruses. So in this blue box is our Australian lineage H5s. And I'm going to draw your eye to those that are highlighted in blue. And these are H5 viruses that were detected in turkeys in 2020. And so it was this group of viruses which I wanted to study further for the capacity to evolve into highly pathogenic forms. Then also, recently in 2023, there's been the detection of H5 viruses in Australia. However, the HA gene is slightly different. It actually originates from that Eurasian lineage that I described earlier. So I also want to examine the biosecurity threat of a virus from this newly detected lineage in Australia, HA lineage. So the experimental approach to testing this will characterize the ability of wild-type Australian lineage H5N2 LPAIV to infect and replicate in chickens. So as discussed in the phylogenetic tree, we'll have the Australian lineage H5N2, which was detected in turkeys. We'll also test the recently detected H5 LPAIV that has the Eurasian lineage hemagglutinin gene. And then as a control of sorts, we'll test an American lineage H5N2 in birds. And this is from a lineage that's known to convert from low path to high path. And what we'll do is we'll monitor clinical disease. We're going to quantify virus shedding from the oral pharyngeal and chloral routes. We'll also quantify virus replication in numerous tissues. And that comes back to that question, is the virus capable of productively infecting and replicating in poultry? And then through this study, we'll actually sequence virally infected tissues to see if we can detect any potential changes in the genome that's associated with high pathogenicity. So this is part one of the project, providing a holistic overview of the ability for these viruses to productively replicate in chickens. And in the second part, we're actually going to dig deeper and dig further into the ability of our Australian H5 LPAIV to convert to highly pathogenic forms, because it's a big unknown. Can Australian lineage and H5s detected in Australia support the genesis of high pathogenicity? So to address this question and this unknown, I'll use a technique called reverse genetics. And this is a tool where we can clone the virus genome. So we can take a copy of the virus genome and we can introduce that using our molecular tools to generate an infectious clone. And we can also manipulate the genome to incorporate the pathogenicity determinant. So the predominant one is that polybasic cleavage site motif. So it has that strings of R's and K's, that basic amino acids, which will incorporate into the genome of these low path viruses. And we'll actually test in chickens to see whether or not these viruses are able to support a highly pathogenic phenotype, or whether other changes are also required. So which cleavage sites will I introduce? So in this table here, I've highlighted in red boxes cleavage sites from viruses that were sequenced from various outbreaks. And I'll be generating reverse genetics viruses, and then I'll be incorporating these known virulence determinants into the HA gene of our low path viruses. And we'll characterize to determine whether or not the virus does display high pathogenicity or not. And we'll do this via advanced cell culture systems. And we'll also do animal infection studies to test the pathogenicity in chickens. So the advanced cell culture systems that we'll be utilizing to examine whether or not the viruses are able to display a highly pathogenic phenotype will be generating chicken neuron cultures. So as described previously, highly pathogenic avian influenza virus can replicate systemically, including in the central nervous system in the brain. So once it is able to get to the brain, it replicates profusely and the outcomes are dire. So what we can do is from chicken or duck embryos, we can actually harvest the brain and culture the neurons. And from there, we can infect the neurons with the viruses, so the wild type or the engineered viruses, and see how they performed. And this will be an indicator to us. Does this virus have the potential to replicate in the CNS, and can it potentially replicate high path infection outcomes? So this is one tool that we'll use in our assessment. We'll also have other tools in our assessment. We'll then move into reverse genetic studies of avian influenza viruses in poultry. So this is, again, we will follow the same principles as the wild type viruses, the viruses that aren't engineered. So we will infect chickens, we'll monitor clinical disease. Do they perform like a low path, or do we see disease symptoms reminiscent of a high path? We'll quantify virus shedding for the oropharyngeal and cloacal routes. We'll also quantify virus replication in numerous tissues. So is it replicating systemically, indicative of a high path? Does it cause severe disease? And we'll also sequence these virally infected tissues to determine whether or not we can detect changes in the genome associated with high pathogenicity. And I've incorporated this aspect because in previous studies, when infecting chickens with reverse genetics clone, we've seen that populations with even more basic amino acids have been favoured, so the virus has mutated again. So we do want to keep an eye on that population to look at the dynamics and see what's happening there. So through this project, we hope to provide some numerous industry relevant outcomes, including enhancing poultry industry preparedness and resilience through a deeper understanding of the risk profile of Australian-lineage H5 LPAIVs. Potentially, we can guide optimal sampling strategies for early detection of H5 high path. So if we see in a particular tissue type that we can detect changes that are reminiscent of high path avian influenza viruses, we can then guide to say, target this tissue type. This is maybe where the selection pressure is happening. We can potentially increase resilience through development of an Australian relevant vaccine seed strain, and ultimately enhance understanding of about biosecurity threats. Again, just touching on this again, our outputs are a deeper understanding, and the outcomes are really dependent on the research results. So like I said, if we do detect changes that are associated with high pathogenicity, this can in certain tissues, this can optimize our sampling guidance for early detection in flocks. Also, conversely, if we find that by inserting the cleavage site motif, this virulence determinant, into our Australian lineage H5s do not support the emergence, this suggests there may be other key factors that are involved. And then it also aids us in identification of candidate vaccine virus for use in local markets to complement current outbreak response strategies, because we've demonstrated that it is unlikely for this virus to convert to highly pathogenic forms. So the researchers involved with this study is myself. I'm principal researcher, so I'll bring influenza pathogenesis expertise, influenza reverse genetics, and cleavage site motif expertise. And I'm collaborating with Dr. Frank Wong on this project. So here is the Australia's Wawa Avian Influenza Reference Laboratory expert, and he's a co-investigator. He also brings expertise in genetic and antigenic evolution. And I also want to highlight, as part of this research, we'll be recruiting a research technician. So we're highly excited to bring someone on board to be able to deliver these critical research. And then we will be supported by animal studies team, agent characterizations, histologists and pathologists to really deliver a holistic overview of the biosecurity threat of our H5 viruses. And with that, I want to thank you and open the floor to any questions if you have any. I'll stop sharing now.
Wonderful. Thank you very much, Jasmine, and providing the audience an insight into the work that you've just commenced. An exciting couple of years ahead. as Jasmina said in the closing, more than welcome to take any questions from our audience today. We have the Q&A chat or Q&A function available to use. You can answer your question, ask your questions there anonymously if you wish to. You can also take yourselves off mute to ask questions. And whilst in webinar mode, I can't see all of your faces there. I've been advised that you can. So if you wish to take yourself off mute, more than welcome to if you feel comfortable doing so. Whilst our audience is hopefully drumming up a few questions, there is a question that has come through in the Q&A about individual animal survival with respect to the high path strain, which Michelle has answered there. Michelle, I might open it up to see if there's anything further that you wish to add to the response there.
Yeah, so the question was about individual mortality in animals due to the strain, and what about people? And the answer is that kind of depends. So if we think about wildlife, we don't have a great idea of what is happening in the vast majority of species, but we are seeing, so for example, in sandwich terns, 16% of the adults died and 100% of the chicks died in 2022. Like, it was really devastating in the species. But when you do animal experiments with mallards, even young mallards that have no antibodies, at least 40% of them survive. So there is really big differences. In poultry, I think the virus is extremely devastating, but of course, due to sort of the culling response, I'm assuming it's 100%, but we won't know. In humans, the case fatality rate varies very much dependent on which lineage humans are infected with. So if it's this 2344B strain, it seems that cases in humans are extremely mild. Many people just get conjunctivitis or influenza-like illness and survive. But those older lineages that are circulating in poultry in Asia, like 2321C in Cambodia, the case fatality rate is between 40 and 60%. So they're actually quite severe infections. So again, the answer is it depends.
Thanks very much, Michelle, and I hope that has been informative to the asker of the question and to others on the call. Just having a look at the Q&A, they've got nothing else that has come through. So what I might do in the interest of time and maybe being able to let everybody out earlier from the webinar is I'll move into sort of the wrap up of the webinar, but would like to let anybody know if there's any questions that you have or that come to mind afterwards. I know in the chat function we've shared Michelle's email address, or if there's any other that you wish to ask of the program of Jasmina's work, feel free to email the program. Some of you may have my AgriFutures email address. Otherwise, you can email chickenmeat@agrifutures.com.au. We'll provide all of this information together for you guys and ensure that you get responses to the questions. So in closing, I would like to bring your attention to a survey that will pop up on your screen very shortly, or as I'm speaking. Yeah, there we go. And this, we would greatly appreciate your time in just providing a little bit of feedback, by the way, responses to these questions. So that's just to make sure that we can make improvements to upcoming webinars, part of the series, that the content that we're providing is informative, is valuable, and we're always open to take suggestions on webinar topics that would be of interest to you. I'd like to extend a big thank you to our speakers for today. We greatly appreciate your time and generosity in sharing your knowledge. And I'd also like to thank everybody that has registered and joined the webinar today as well. As mentioned at the start, the webinar has been recorded and we will make that recording available online, and we'll share links to any resources or items mentioned that are of relevance. So with that, thank you very much once again, and we hope to see you all at the next webinar. Thank you very much. Goodbye.